This presentation entitled Introduction of Next Generation Sequencing in Clinical Diagnostics is presented by Dr. Rong Mao. She is the Medical Director of Molecular Genetics and Genomics at ARUP and is an Associate Professor of Pathology and Co-Director of the Clinical Medical Genetics Fellowship Program at the University of Utah School of Medicine. Dr. Mao received her MD from Capital University of Medicine in Beijing, China, and her MS in Molecular Pathology from Beijing Union Medical College. She is certified by the American Board of Medical Genetics with a subspecialty in clinical molecular genetics and by the New York State Department of Health with a subspecialty in genetic testing. I'll now turn our presentation over to Dr. Mao.
Well, good afternoon. Thanks for joining today's webinar. I'm going to talk about the next generation sequencing in clinical diagnostics. Here is the objectives for today's presentation. I'm going to describe the methodology of next generation sequencing and compare different platforms, discussing the processes, validations, and use of the gene panels, exome sequencing. I'm probably not going to touch base with the whole genome sequencing because it has not been used in clinical diagnostic yet. And then I will talk about understanding and appreciate the complexity of the data created by next generation sequencing. We need to go back to the early stage when we do the mutation testing. Sanger sequencing is considering the gold standard for mutation detection, and this technology has been invented in 1977. Is to using the chain terminating inhibitors technology to be able to sequence our genome. And the products will be run on the polyacrylate gel and exposed on the X-ray film. And in old stage, we're reading the sequence from the bottom to top, goes through the different nucleotides. And in 1986, then the fluorescein Sanger sequencing technology has been developed, and through capillary electrophoresis will be significant increase the throughput and the quality of Sanger sequencing. And here is one of the examples for Sanger sequencing results seen in mutation severe softwares. On the top is the reference sequence, and here is the actual patient's sequence from the forward direction. On the bottom is the reference sequence again, and here is the sequence from reverse direction. In clinical laboratory, we sequence the samples from both forward and reverse direction. That's increased the accuracy of the results. And Sanger sequencing now is the matured chemistry with good quality of Sanger sequencing. They can give you roughly 600 to 800 base pair read length. And in each Sanger sequencing peaks, it counts more than 1,000 equal length termination products. Bidirectional sequencing will be increase the accuracy and is very well established base calling algorithms. And then in 2005 is a new technology has been developed, and this paper has been published in Nature talking about genomic sequencing in the microfabricated high-density picotitre reactors. And in this technology, they'll be able to have millions of mini sequencings occurring at the same time. It has been called massively parallel sequencing, and this will be significant increase the throughput, allowed us to be able to large scale sequencing our genome. Compare with the Sanger sequencing, then this technology has been called next generation sequencing. And since next generation sequencing has been developed, then we are in the paradigm shift. From Sanger sequencing, now we switch to the next generation sequencing for the human genome. Here is a very brief workflow for the next generation sequencing. Genomic DNA will go through the fragmentation till the fragments will be suitable for the next step sample processing. The fragmentation can go through either sonication, nebulization, and sometimes you can use restricted enzyme to cut the genomic DNA. Then the DNA will be enriched for the targeted region. It can use in long-range PCR, emulsion PCR, or array capture. Only focus on the targeted region you are interested. Then these products will be followed by the next generation sequencing library prep. The products after this will be loaded to the next generation sequencing instrument for the massively parallel sequencing. Then after the data has been generated, will be through the bioinformatic pipelines have a data analysis. And here's a little bit details for the library prep. When you have your DNA fragment, first step, the exon nucleus will be blinding to fill in the gap at the 3' and 5' of the DNA fragments. Then go through the phosphorylation to add on phosphate on the both end. Then it will be add on this A overhang. This will be help go through next step, have a ligation of the adapter on both ends. And we are going to using this for next generation sequencing. The Roche 454 is one of the early platform. Here is the workflow for this instrument. The sample goes through the fragmentation, and then the library prep will be add on the adapters on both ends. Then this DNA fragment, the adapter will be attached to the beads. And once the DNA has been diluted further enough, each bead is only capture one DNA molecules, and then followed by the emulsion PCR. In the emulsion PCR will generate a clonal amplification for this single DNA molecules. After emulsion PCR, the emulsion will be broken. The products has been loaded to the picotiter plates. On this plates, there's millions of little wells. Each wells only allowed one bead to sit in, and this will be go for the next generation sequencing. And for the Roche 454 sequencing, they're using pyrosequencing chemistry. The the DNA templates is immobilized, and the solution of AGCT nucleotides are added subsequently. When there is a match, then the pyrophosphate is released, and ATP sulfurase will be converted pyrophosphate to ATP. Then the ATP is going to be using as the energy source, will be allowed the luciferins mediated conversion of this luciferin to the oxyluciferin, then will be generate visible lights. And the lights produced in this reaction is detected by the camera, and the sequencing can be analyzed. And this is the Roche 454 platform. This is a large scale instrument, and then there's a small scale instrument is also available. Now the more popular instrument is Illumina instrument series, and here will be the workflow for these platforms. The library prep will be similar as the Roche 454, however, will be follow Illumina library prep protocol. After the library prep, the DNA products will be loaded to the flow cells. I'll show you later. On the flow cells is a glass surface. It has been spotted millions of these anchor sequences. The anchor sequence is a paired. One of them is have a sequence complementary to the 5' adapter. Another anchors have mass sequence match with a 3' adapter, once the DNA product has been loaded to the flow cell surface, then the DNA will be hybridized with 5' anchor and 3' anchor will be formed the DNA bridge. Then the DNA cluster will be generated through the bridge amplification. After this experiment, then the sample will be denatured and cleaved from one end. Then the sequencing will be go through this direction. Here is just showing the workflow. And the Illumina chemistry is sequencing by the di-terminator sequencing. Once the primers anneal to the target, then the 4 reversible di-terminator NTPs has been added to the reaction. It's only one nucleotides have the sequence match with the templates will be incorporated, then this signal will be captured. After this signal has been captured, then this terminator and the dye will be cleaved. Then the DNA strand will be able to go further extension. Then the reaction go to next cycle. If we go 100 cycles for the sequencing, we'll be able to sequence 100 base pairs. And here is showing you the Illumina flow cells. There's 8 lanes. You can load your samples to the 8 different lanes. And the signal has been captured by this high resolution CCD camera. And one important sequence technology is the pair and read sequencing. It will be sequenced 100 base pairs from one direction, then the DNA templates will be flipped, then you will be sequenced from another direction. At the end, you will have 100 base pairs from 5' end and 100 base pair sequence from reverse directions. The advantage to doing the pair end sequencing, this will be increase the read coverage per cluster. It is more accurate reading and alignments. And the more important is pair end sequencing will detecting small and large insertions and deletions and inversions, and also other large gene rearrangement. Here is the Illumina instrument series. There's a high throughput instrument, it's a HiSeq 2500. With each run will be give you 1,000 gigabase reads, and within the 125 pair and reads will be take 14 days, and it will be cost the reagents per run is around $14,000. In addition, there's a 2 smaller scales instrument. They can give you the different throughput, which allows you to have the flexibility to choosing which platform will be best fit for your laboratory. And next generation sequencing are data rich. The imaging capture are terabytes files. This imaging captured files will be go through the imaging processing, will be convert imaging to the sequence basis, and then generate a sequence read files. Files will be able to be comparable to the downstream software's program to have alignments and variant calls. And there's some important parameters for next generation sequencing you need to be aware of. First, we're talking about alignments. For human genome sequence, we aligned the sequence to the human genome reference sequence. Now it's the HG19, but the newer release, the HG20s through the gene bank is already available. Another important parameter is the coverage. The depth of coverage will be allowed you to know whether you have reliable sequence results from your next generation sequencing. And here is one of the example. In this particular position, the coverage for this position is 184 X coverage, which means for this particular position, it has been sequenced 184 times. And in 2008, there's a study has been come up and showing in order to obtain reliable sequencing results, you need a minimum 15x coverage. If there's a nucleotide substitution, then there's another important parameters to be known is the base change percentage. By the deployed human genome, you are expecting 50% in the base change percentage. However, that's the ideal situation, and in many situations, you can have could be 60% to 40%, and sometimes can be 30% to the 70%. And the Sanger sequencing conformation has been widely used in clinical laboratory for the variants. They are either pathogenic mutation or variants unknown significance, or even the benign sequence variants has been detected by the next generation sequencing. Then any report in going to be any variants going to be included in the clinical report. They are go through the Sanger sequencing conformation. And here is the sequence we just saw previously as a deletion G, and this has been Sanger confirmed. And since next generation sequencing technology has been developed, you can see the sequencing human genomes price has been significant drop. And 2001 is a complete human genome sequencing project. At that time, if you sequence one human genome, it cost $100 million. 2007, just right before next generation sequencing technology invented, you still need to using $10 million to sequence one human genome. And since then, you can see the price has been significant drop. To date, if you sequence one human genome, it's cost $4,000. And next generation sequencing has been established a new landscape for genetic testing. We can do single gene diagnostics, multi-gene diagnostics, exome sequencing, and optical genome sequencing, and the complexity has been increased with the different tests. And now I'm going to talk about multi-gene testing for the using the NGS testing. So the multi-gene panel diagnostics is fitting well for the genes. They are very large, difficult for Sanger sequencing, no mutation hotspots. So you basically need to sequence through all coding region of the exons. They are very expensive by Sanger sequencing. Or the conditions have multiple genes responsible for the same phenotype. They are genetic heterogeneity, and it will be very difficult to sequence multiple genes by Sanger sequencing. Or the conditions with phenotype overlapping conditions, then there's many different diseases overlapping, and you need to testing them all. But all of these conditions can be fit in well for the next generation sequencing multi-gene panels. And here is some of the example for the multi-gene panel. For example, mitochondrial disorders. We can sequence mitochondrial genome up to 100 disease-causing nucleus genes at the same time. Cardiomyopathy, there's 85 genes known, of course, responsible for the condition. For the X-linked mental retardation, there's 95 genes. And I'm going to talk about mitochondria disorder and using this as a model disease to talk about the multi-gene panel. Just give you a little bit background for the mitochondria and the mitochondria disorders. Mitochondria is a microorganelle located in our human cell's cytoplasm. The function of mitochondria is converted substance from our food into the energy source, which is in the form of ATP. And ATP is the energy source for our human body. And it is well known there's more than 1,000 genes. They can be coded by the nucleus gene or can be coded by the mitochondria DNA. They have the functionally involving for the mitochondria. And the mitochondrial genome is another genetic inherited materials besides the nucleus DNA. It is a double-stranded circular DNA, has a length of 16.5 kb. The mitochondrial DNA, majority of them, they are coding genes. They don't have introns. It also lacks the histone and the DNA repair mechanism, so there's a high percentage of the mutations within the mitochondrial genome. Overall, mitochondrial genome has 37 coding genes. They coded for 22 tRNAs, 2 for RNAs, and there's 13 genes coding for the short peptides. There's one important concept that need to talk about here is the heteroplasmid level of mitochondrial disease. When there's a mutation occur, then there's a mixed population of mitochondrial genome. There's a mutant mitochondrial genome as well as the normal mitochondrial genome. The percentage of the mutant mitochondrial genome is being called heteroplasmid. And the heteroplasmid level have to be higher enough in order to causing disease. In many situations, the heteroplasmid level need to greater than 40% in order to develop mitochondrial disease. And the mitochondrial disease is maternal inheritance. Besides gene coded by the mitochondrial genome, there's many nucleus genes is also coding for the protein they are involving for mitochondrial functions. If there's a mutations in the nucleus gene, then they fall within the Mendelian inheritance, such as dominant recessive or X-linked inheritance. And here is a very busy table. It's showing there's a 108 nucleus genes. They are well known to causing mitochondrial disorder. And some of them are the most common ones to causing this condition. For example, POG, TAS, SURF1, and MPB17. And the disease frequency for mitochondria is 1 in 5,000 live births. 20% of the diseases caused by the mutation in mitochondria DNA, whereas majority of them is caused by the nucleus DNA mutations. In order to have a comprehensive DNA testing, then the next generation sequencing will be the best platform to be able to detecting for this condition. So multi-gene panels has been developed using next generation sequencing technology to be able to sequence mitochondrial genome as well as 108 nucleus genes at the same time. And in order to accomplish with large deletion duplications, then the high density array CGH will be able to detecting the large deletions and duplications, but we're not going to discuss this in this presentation. For mitochondrial genome next generation sequencing testing, it's well within the library prep protocols for the next generation sequencing protocol. The only difference is the upper front DNA enrichment. Mitochondrial genome is using long range PCR to do the DNA enrichment. It has 5 pair of primer sets. The 2 blue ones will be one of the 2 pair of primers will be sequence, will be amplification for the mitochondrial genome once, and then the green will be another 3 primer sets will be amplify the mitochondrial genome again. The reason to doing this is mitochondrial genome is highly polymorphic. There's a lot of polymorphisms can causing the primer sites allele dropout. The different primer locations will be able to avoid allele dropout for the long-range PCR. And on the left-hand side is the Argos joint to showing the PCR products. And the nucleus genes enrichment is using the limbo gene targeted DNA enrichment technology. The genomic DNA targets will be go through the fragmentation and followed by the next generation sequencing library preparation protocol. Adapters has been added to the DNA fragments. Then we'll go through the hybridization stage. The hybridization stage, you have the library base. All of the library bases have the sequence complementary to all of the exons of 108 nucleus gene. Then the base will be hybridized to the complementary DNA sequence, and the bases are the biotinidase labeled. Once the hybridization is completed, then the biotinidase will be hybridized to the streptavidin-coated magnetic beads. Then the products will be captured. Then the unhybridized products has been washed off. Then followed by the amplification, the DNA then will be generate the DNA products will be able for the next generation sequencing. And here is showing you all of the workflows for next generation sequencing from the day ones of the library prep until the data is available for the review. It's take about 2 to 3 weeks. So it's a very long processing. And here is showing you the mitochondrial genome sequencing results. On the top is the linearized 16.5 kb mitochondrial genome sequence, and this is the 37 genes. Here is the actual sequence results we got from next generation sequencing. And in this particular position, you can see we have 748 depths of coverage. And here is one of the example for the mutations in mitochondrial genome has been detected. It's a G to A substitution at position 3460. This is resulting a nucleotide resulting an amino acid substitution is alanine changed to serine. Is in the gene called ND1, and this is the mutation causing mitochondrial disease. And here is one of the examples to showing you the mitochondrial nucleus gene. Here is one of the nucleus gene, and all of these bars are the exon region. And here is the actual reads cover for the coding region of this gene.
Okay, I'm going to present a clinical case. This is a newborn patient. Patient has abnormal newborn screening with elevated phenylalanine, and then the follow-up plasma amino acid showing elevated tyrosine and methionine. Then he had a significant fetal strife and feeding difficulty and fat malabsorption. Then he had a liver failure and transplantation at 7 weeks of age. Subsequently, he developed hypotonia and psychomotor regression and died at 23 months of age with cardiac arrest. And we fortunately get a tissue sample from these patients. So we did mitochondria genome sequencing as well as the 100 nucleus genes with mitopanel. And we did identify 2 mutations in the DGUOK gene. And here is the reference sequence. And a nucleotide substitution has been identified at the position 591G to A change, which is a silent mutation. However, this is the last nucleotide of this exon, which is affected for the normal splicing site. So this nucleotide substitution is causing splicing site defect. Here is to showing the second mutation, which is a 2 nucleotide deletion causing frame shift mutation. 2 mutations identified in the DGUOK gene, which has confirmed the diagnosis for the autosomal recessive mitochondrial disorder caused by the DGUOK gene. We're going to talk a little bit about clinical utility of mitochondria gene panel. This panel will be very useful for the differential diagnosis, confirm the genetic etiology of the disease. Specific disease-causing mutation identified will be helping for the family risk consultation and the testing for the other family members. And confirm the diagnosis will be helping for the patient's management and treatment. Now we shift gear from gene panels and talk about exome sequencing. We have 2 tests available for the exome sequencing. The exome sequencing with symptom-guided analysis testing, which is including proband plus up to 5 family members with same price. And this testing allowed with testing probands as well as family members like parents samples and other family members, affected or unaffected individual. We also provide a proband-only test. This test turnaround time is 12 to 16 weeks, and the specimen type can be peripheral blood or other sample types is also acceptable. So what is a human exome? Exome is the portion of the gene or genome that codes information for protein synthesis. It has been estimated within our human genome there are 21,000 genes comprised of 180,000 exons. And within the 21,000 genes, there's about 4,000 genes. Mutation within this gene causing genetic diseases. They are Mendelian inheritance, but can be also mitochondria maternal inheritance as well. Then when we do exome sequencing is a focus on the part of genome we understand the best. And exons comprise 1% of genome. However, 85% of all known disease-causing mutations are located on exons. And exon sequencing price is only 1/6 of the cost of a whole genome sequencing, so exome sequencing will be more cost-effective. Then you have to keep in mind for exome sequencing, some region will be missing by this test. For example, some protein coding genes, they have a pseudogenes that will be interfering for the assay. And some exons have a high GC rich region, can have the capture probe drop off, or the non-genic control elements, copy number changes and structure changes, etc. Exome sequencing is a very powerful tool for gene discovery. The last couple years, there's over 200 genes has been discovered. Exome sequencing is provide the sensitivity of the test about 25%, And this is provided by the NIH Undiagnosed Disease Program. By the best selected cases, then the sensitivity of exome sequencing is 25%, which is consistent with ARP clinical exome sequencing sensitivity. And exome sequencing is provide diagnostic odyssey, as especially for the patients with multiple congenital anomalies, intellectual disability, and some unexplained developmental delay or declining. One of the sequencing strategy is recruiting multiple family members for the test. That will be significant increase the yield of positive. Here is showing one of the families have multiple family members affected for the same condition. Once we exome sequencing for the two affected individual, then when you do the data analysis, you can focus on the variants shared between these two affected individuals. If we recruited one more affected family members, then we can more narrow down to the variants will be shared between three of them. And here is the brief processing for the clinical exome sequencing. Samples will be indexed and pooled together for the sequencing. Then the exome region will be captured by either Agilent or Nimble gene liquid capture assays. Then the sample will be run on the Illumina HiSeq instrument. Once the sequencing done, then the data will be extracted, followed by the alignment and variant calling by the bioinformatic pipelines. Then the candidate mutation variants will be analyzed and the candidate list will be generated for the Sanger confirmation. Then the results will be completely interpreted and reported out. Here is showing you the workflow for the exome sequencing. Once everything works perfect, and from the day one we start in sample prep until the days we have a data analyzed, it's probably take at least 3 to 4 weeks. And this will be the exome capture assay. Here is showing the Agilent exome capture, and this will be similar as the Nimble gene capture we described previously. And this will be showing you the exome sequencing on the HiSeq instrument like we showed previously. Here is showing you the exome interpretation algorithm has been developed in ARUP. Every exome you will be obtain 20 to 30,000 variants. Once you filter out the common polymorphisms as well as the synonymous changes, you can narrow it down to about 2,000 variants. Then you will do multiple analysis for the data. Then you should apply the inheritance pattern analysis, de novo analysis, or looking for the variants in the gene has been documented in the HGMD or OMAM, that will be known disease-causing genes. And all of the variants will be interpreted based on the population frequency, disease-specific database, computational prediction program, as well as AREP frequency. And the variants will be interpreted based on the patient's clinical symptoms. After the data has been completely analyzed, candidate variants will be generated, then will be sent for Sanger confirmation and results will be reported out. Here is the ARUP bioinformatic pipeline. I will show you some function of this program. You can apply the population frequency filtering and then excluded the 5' UTR or deep intronic region, filter out the low quality reads, and also apply for the computational protein prediction program. You can also enter the genes and regions if you're interested, or only focus on HGMD or OMAM genes. We can also doing the pedigree analysis with this program. If you are looking for the variants, you can go through the IGV viewer And we also can only analyze the incidental findings for the 56 genes. Here I'm showing you one of the example for AREP frequency, and you can see with this variance, we saw total 42 times in our exome samples, and they are on the different genetic condition. This information will be indicating this is a common variance will be seen in the patients or even general population. This is the most likely benign. So what is the incidental finding? Incidental finding is to see the variants found by exome genome sequencing which are unrelated to disease of interest. Majority of them, they are benign. However, a small number of them might be well described and disease-associated mutation.
Therefore, in 2013, American College of Medical Genetics released a recommendation for reporting incidental finding in clinical exome and whole genome. Within these recommendations is to talk about laboratories should return with each genomic sequencing order results from 56 genes in which the mutations greatly increase risk for 24 serious conditions but treatable disease, even if the clinician does not suspect the patient have them. And the ACMG working group is also recommended laboratory active research for the specific type of mutations in the specific gene listed in this recommendation, and here listed the conditions. And since the incidental funding recommendations has been released, then there's a lot of discussions and critics, and especially for the children's we did exome sequencing, there's a lot of concerns to release incidental funding for the adult onset condition. Therefore, in 2014, ACMG has revised the recommendation, allow the patients to opt in and opt out for receiving the incidental findings. And exome sequencing is the really evolving processing. It will have lots of communications between the laboratories and the physicians. Once the testing has been ordered, then our genetic counsel starting discussion the cases with the physician to obtain the clinical phenotypes and also the laboratory testing results, family histories of the conditions. All of the informations will be helping for the future data analysis, and also is helping for the physician to consenting the patients and their family members. Then there's a lot of evolving for the data analysis. I'm not going to go through the details. And the post-analytical, once the report is ready, we are follow up with the physicians to discussing the results and the limitation and the quality of exome sequencing. And here I'm quickly give you one of the examples for the exome sequencing. This is a patient's 7-year-old male Caucasian. It's suspected for the disease called mucopolysaccharidosis or cranial de Lange syndrome. The patient's neurological finding including delayed speech, fine motor delayed, cognitive delayed, dysmorphic features including hirsutism and court face. Patient has short stature, overweight, skeletal finding including diffusion, osteopenia, significant delayed bone age, craniofacial finding including cleft palate, macrodentia, and cleft earlobe. And dermatology findings has the eczema. And the laboratory testing showing metabolic workup, normal mucopolysaccharidosis screening, urine organic acid has non-specific elevations, PAA was normal, patient has a normal karyotype, microarray, Fragile X testing, and the 22q deletion testing was normal. Then the brain EEG suggests non-specific diffusion cerebellar dysfunction. MRI demonstrate a mild cerebral atrophy and also changes comparable with the diagnosis of cerebralitis. And there's no family history in this patients. Then we did exome sequencing for the proband specimen and the parental samples available for Sanger confirmation. We obtained 59,000 variants. Once we go through the quick filter for the common SNPs, then the variance has been narrowed down to the 2500s. Then we focus on the coding region of the genes, then the variance has been further narrowed down to the 754. Then we applied inheritance patterns, HTMD match genes or HTMD genes for the data analysis. We first look at the genes can cause cranial delunga and mucopolysaccharidosis genes. There's no mutations has been found in all of these genes. Then we found a mutations in the SATB2 gene. This is a gene has a mutation in the splicing site. This T to G change will be disrupt the normal splicing site. As a result, will be spliced to the next splicing site. As a result, in the transcript will be including additional intronic sequence. Usually, this will be generate a premature stop codon causing truncated protein. And this variance, we did a Sanger confirmation on the probands, and we're also testing on the both parents, and both parents have been showing they don't have this mutation, which means this is de novo mutation. And here is the information of this gene. We don't have enough time, so I wouldn't go to too much more details of this information of this gene. The function of this gene is involving for the transcription regulation, chromatin remodeling, and played important roles for the craniofacial pattern and the brain development. And there's a report for the de novo translocations involving for this gene. It's associated with a patient has isolated cleft palate, and more report indicating these mutations can causing the isolated cleft palate, generate osteoporosis and a profound mental retardation. So all of this is fitting well for our patient's phenotype. So we reported this is the pathogenic mutations causing patient's phenotype. And in this case, we do not have the incidental finding. And before I finish up for the presentation, I will give you a little bit information about the regulation and assay validation. So since next generation sequencing has been used in the clinical laboratory, then the CAP already developed the CAP checklist for the next generation sequencing. And ACMG laboratory QA committee developed the clinical laboratory standard for next generation sequencing, and the CAP proficiency testing now is available. The first shipment will be 2015. This will be the methodology PT, is not for the specific analytes. Here I'm going to share very briefly for the assay validation. For the NGS validation is more complicated than the others because there are lots of informations. And there's still the two parts of the validation. One is the accuracy, which is to determine the concordance of results in the two different assays. The samples using for accuracy will be the samples with the new mutations or the correlative repositive samples. This HapMap sample NA12878 has been widely used for the assay validation because it has been deep sequenced many times. There's a HapMap results for this sample as well as ARRI data. And for the accuracy, We needed to determine the analytical sensitivity and analytical specificity, and for the position, we need to do the within run and between run. Within run will be determine the concordance of the variance of the triplicate in one sample, and the between runs will be the concordance of the variance costs and the qualities between the different runs. And here is one of the samples to showing within run. One sample has been tested in triplicate. You check the concordance of the variance. You can see majority of them, they are concordance. And here is a between run is also showing the status well concordant. And NGS validations also need to provide a reportable range and reference interval, and it will be give you the list of the genes and reference and the regions analyzed, and the reference range will be the common polymorphism found in the normal samples. So to conclude today's presentation, next generation sequencing technology provides opportunities for large-scale genome sequencing. The complexity increased from gene panels to exome sequencing to whole genome sequencing. Next generation sequencing requires advanced information for data analysis, and annotation tools are rapid advancing. Variance detections need confirmation, and the causality is need evidence. Clinical and family information is critical for assessing significance. And next generation sequencing technology will be going to have a huge involving, and this is really a teamwork. And you can see we have R&D and bioinformatics, clinical laboratories, genetic counselors, and medical directors to form a large team to be able to implement this complicated technology into the clinical laboratory. And I will stop here. Thanks very much for your attention.